Good morning. Studying relation about how DNA. Point of the session, I don't know. Not working. No. Okay, thank you. The location determining transcription binding site when another is not amount, perhaps it the nearby gene. We've been interested in organization specific in vitro, not all aspects in the DNA sequence. That was transcription that are recognized by corresponding transcription and on in vivo. But if you look at these sites already in the in vitro map where transcription factor itself is not present, then for most transcription factors at least, you see the following picture, which is on average nucleus on the functional binding sites in vitro and in vivo, pretty levels. Of course, there are exceptions to this. This is on average for at every site, and also a couple of transcriptions this rule. But by and large, we see this picture already in vitro. Perhaps the DNA-coded nucleosome organization facilitates transcription factor and consequently can therefore have influence on expression outcome. So, in asking what is it in the DNA sequence, what are the key features that actually may be responsible for doing this? And several reasons we believed that these poly-A sequences, which are highly abundant in eukaryotic genomes, may have a big role in this. And that actually is one reason, just their sheer abundance makes likely candidate. Second, we and many others before us, poly-A sequences strongly favor nucleosome formation. Very nice work by Kornberg and Stryer dating over 20 years ago have some boundary for nucleosome formation, not just over the boundary itself, but also when it's meant to have a binding site for a transcription factor, it will also enjoy created by the poly-A element, and therefore may be happens instance of the same transcription factor across these poly sites near it. And so that was a final prediction, which is that if you now go to a promoter site near a binding for a factor and you deleted that element on the nucleosome occupancy, which would go up site and consequently would more inhibit and reduce expression, and exactly was shown in GI over 15 years ago in East. And so we were intrigued by predictions and controlling and regulating transcription gene expression, and we set out to set us with a tool in a more comprehensive. I will describe the experimental system to show you some results that we obtained from it. So we designed experimental system in yeast, which we have one strain in which at a fixed chromosomal location we put a promoter that drives the expression of a red fluorescent protein, or RFP. At the same time, we have YFP, in front of which we integrate then different promoters we want to test. And with these strains, we can measure the dynamic gene expression behavior of these various strains, controlling for genomic context effects because all place in the same chromosomal location, post-transcriptional effects because promoters are hooked directly into YFP without any without their. Most importantly, by having this system, we can distinguish expressions that are as small. So because all of these parameters are controlled, then that we see between genes in which we have different promoter sequences only to differences in the integrated most ideal system we could think of for studying the mapping. So once we generate these libraries of promoters, we then measure them in 96 well plates, inserting a different strain with different promoters to every well, and then tracking the growth, RFP, and YFP of these populations, starting from a small number of cells in different growth conditions. So here's for one library of 96 promoters. Here are different growth curves. You can see they're pretty much identical because these strains are essentially wild type, except for the in front of the YFP. Similarly, they all drive RFP using the same course you would expect. Indeed, find that the activity of the RFP is between all strains, which also shows you the experimental variability of the system. And finally, have different promoters driving YFP and a much larger range of. We believe that when we see these in the library, they are attributable to differences. So we're generating many different libraries using. They mainly is a library of synthetic using starting with a promoter that Struhl and Vishayer studied 15 years ago. But now making about 80 or 90 different sequence variants of this promoter to address several different questions related to the possible effect of DNA-encoded nucleosome organization, more specifically poly-A sequences, on gene expression. And the first question we asked is whether both boundary elements, both poly-A elements that appear in this promoter in its native form actually matter for transcription. And so what I'll be showing you in the next slides is basically different promoter variants that we generated and their corresponding YFP activity levels across time as the cells are growing. These are going to be these curves of YFP activity, and you'll see here the different promoter variants. So this is first the wild type promoter with the single binding site for the transcription factor GCN4 flanked by two poly-A elements of different length. This is the corresponding activity pattern. If you now delete the left poly-A element, like Eran Segal did 15 years ago, or the right poly-A element, we see that we get significant reductions in gene expression levels. If we delete elements, we almost entirely abolish gene expression. If we now store just the poly-A element but make it longer, we can pretty much wild type levels. And if on top of this strain, we bring back, we can induce transcription even higher. And so a set of variants, you see that by only making changes to sequences, namely these poly-A elements, we can dramatically affect in site for GCN4, largely in that the DNA-encoded organization matters in, to some extent, the organization of nucleosomes in vivo of transcriptional outcome. So our next question, given that these poly-A elements affect nucleosome organization, their effect should be quite general and pretty independent of the identity of the transcription factor that's inducing and also the affinity of the binding site itself. So first, regarding the site, we generated 3 different pairs of comparison where we had either a weak binding site for GCN4, an immediate affinity site which comes in the native form, or it's hard to hear, but a stronger affinity site. And in all of these cases, we considered a comparison in which you either have or do not have this left poly A element. In all cases, we see a significant increase, as one would expect, with the introduction of the to not having the element background. Similarly, we replaced the GCN4 site with either a GAL4 binding site or a site for another transcription factor and measured transcription in the corresponding induced condition of these different factors. And also, like GCN4, we find FO4 that the promoter variant that contains the, as one would expect from a general mechanism that acts in all cases a significant increase in next question was whether effects on gene expression that are conferred by these poly A elements, how do they compare to the level of effect that one can get by changing the affinity of the binding site itself for GCN4? And so this we studied from 2 different genetic backgrounds, one with a weak binding site for GCN4 and another intermediate affinity binding site. And in both cases, we considered upregulating gene expression by 2 different mechanisms. One is making a single pair change to make this weak affinity site into an immediate affinity site, or another way by leaving the site untouched but bringing back the left poly A element and doing the same on this different genetic background. And here are the results that one gets. These are the starting strain, and you can see doing either making the reducing poly A under both genetic backgrounds, we get increases in expression levels, but also at the quantitative levels, similar outputs, suggesting that poly A sequence elements can be as quantitatively speaking as the actual affinity of the description factor. The next question to address is whether the length and the actual perfection position of the poly A element mattered. And this we wanted to ask because of various analyses that we did, in which we found correlations genome-wide between on poly A elements on the genome as a function. Longer poly A elements have stronger nucleosome depletion. Especially perfect poly A elements, you can also have mutations and still get the same behavior, except that the graph will be shifted because you just need to get the same kind of nucleosome disfavoring action that you would store poly A element. And so this we tested by starting with this variant that doesn't have any poly A elements, introducing the left poly A element, a perfect one of length 5, barely see any increase in transcription. If we make it longer to a length of 12, significant increase. Length 17, but with mutations, also a significant increase actually to about the same levels. So indeed, you can trade off the length of the poly A element with its composition, with the number of perfections that it has. And finally, if you further into length 22, you get another significant boost in transcription, as suggesting that indeed this length and composition of the element, which you can, in a predictable fashion, tune the expression level. Our next question was whether the distance poly A elements and the binding site had an effect, and this we thought could play a role again because of the theoretical analyses done by Kornberg and Stryer and some other analyses that we did, in which if you consider the boundary element or the poly A element, which will have this depletion, and it will also it will confer depletion of nucleosomes in its vicinity that will start to increase, nucleosome occupancy will increase as you move away from the boundary. And so the prediction here would be that as you move the element away from the GCN4 binding site, nucleosome occupancy on the fat would start to go up, and correspondingly gene expression would start to go down. And so this is what we explored in the next set of variants, where in addition to the YP activity here, I'll be showing you here just of taking the strongest point on this graph and basically looking at as a function of distance between the poly A element and the GCN for these different variants. So we started with a poly A element of length 20 with a distance of 12 from the GCN4 site. We start to move it away, distance of 20, 80, 100, 65, and you can see kind of as would be predicted, transcriptional outputs to go down and up to a distance of 250 base pairs. Now, for reference that have any poly A elements, and you can notice that at 750, you get a further compared to not having the poly A element. So that seems kind of an outlier to the overall. And so at this distance of 200, we also detect the transcription level compared to a closer and more. It seems kind of adds to the general monotonic impact. Outliers could potentially be very why we went for them, because in those theoretical mentioned before, if you actually continue the graphs, then the predictions of that pattern is actually be periodic. At some point, you have a hyperplan which then acts boundary to what happens next, and so that nucleosome occupancy would go up and then down, and correspondingly transcription factor accessibility. And that's why we generated these strains and made admittedly actual distances at which these things really match our theoretical predictions, and really we need to just more strains that find distances to this question. We also, but I think I can say for certain is that there's definitely a monotonic decay component. You move the poly A site away from the GCN4 site, which is what you would predict. We also generated this different backgrounds that to start with, and in all of these cases we see this general decrease, again with some notable outliers that would be interesting to understand in the future. Then final question that we asked about the system, which in our view most insightful, is actually now based on every sort. It seems that controlling these poly A elements, length, and so on, can significantly affect gene expression in a predictable way. And so we asked about maybe this could be we have so many degrees of freedom, the length, the distances, and so on, by which we could fine-tune gene expression at quite high resolution. What I mean is the following. So here are two promoter variants that differ by a single base pair. And this single base pair change makes the native affinity site, intermediate affinity site for GCN4 into a consensus site. And as far as we know, there's no other sequence change that one can make to get in between affinity for GCN4. And so this single base pair change, here are the differences in activity that it generates. It's over a threefold change in transcriptional outcome. And so we asked the question of what we wanted to get expression levels that are somewhere in between here. So again, as far as we know, there's no other change that we can make to the GCN site that would get it in intermediate and generate that. However, if we from this promoter here, and we now add poly A elements at varying distances, then what is that we can gradually fill in the expression levels in a quite predictable fashion by controlling here the length of these poly-A elements. And so to us is now giving a mechanism at which we can, in which we can actually affect line resolution. And we thought that if we can do that here in evolution, organisms have utilized for fine-tuning gene expression level. And the final evidence for that. So here's different library we created, this time promoters of native promoters. And this is essentially most of the promoters for the ribosomal protein genes in yeast. So we have 110 of them. And something interesting about the evolution of yeast is that at some point there was a whole genome duplication, after which most of the ribosomal proteins actually retained both of their duplicates, but 19 genes lost one of the copies. And so the first question that we wanted to ask, here are the different transcription rates of all the different promoters, just sorted according to their activity. And the first thing that we notice is that if you now highlight where these 19 genes that exist in a single copy are, then you find that promoters, and of course the actual gene doesn't appear in this experiment, you find that their promoters are very strongly enriched to being the most highly expressed, which is what one would expect if the stoichiometry of protein genes is largely one-to-one, then you would expect that genes that are in a single copy would have stronger promoters, and indeed that's what we see, suggesting that promoter sequences have indeed been fine-tuned by the yeast genome for a specific function, in this case for helping in fixing a more proper stoichiometry that you need of ribosomal proteins. Now, of course, the question that we were interested in asking is then, what is special about these promoters that are stronger? What is special about their DNA sequence? And one of the strongest factors we found distinguishing the promoters that are the most strong here is that if you break them up kind of into these three, and you plot nucleosome occupancy as measured in vitro by the model, and actually also as measured in vivo, very striking differences, such that a group of promoters here that have transcriptional activity, they have the strong and closer here to the transcription start site, and everything we know about nucleosome DNA encoded nucleic acid, and indeed found that there's also a much more poly-A element exactly at the region where you see as a strong promoter, suggesting that indeed, at least in one of the ways by which the yeast has evolved stronger promoters for, and again for probably an important biological function by having more poly elements in the specific. So I want to end and just acknowledge the many people. So first, our long-standing collaboration and people of lab, and then people in Malika, the nuclear models, and Elon work on the ribosomal protein. For your attention, be happy to take any questions.
Quick question. Have you looked at the of K-Walt decays or ISCB of strong promoter is in fact found across even the duplicated now retained duplicates, basically in fact change their structure, or is that a structure that was present?
Be a very interesting thing for which we are looking at.
Hi, I have a related. You have looked at the there's correlation as close to the motors, and several different, yeast expression consistently in highly expressed poly-A element.
Yes. So in enrichment of polyelements on average in the yeast, I think population is quite weak with and transcriptional genome-wide as far as the promoters that are highly expressed, like have an overrepresentation of these poly-A elements compared to
genome that is needs an A fusion or promoter weight.
from them that's
90%
or
80 weight. Yeah.
he would have been recently generated that, but we have, so I think that, so I think that would be very, it's possible that a lot of the maps that we were generated from very base composition, different sequences, we just the bias that we have looking at. So
I chromosome level structure cores with entire levels of rich regions, and I was just wondering, particularly because you can have non-perfs and have discouraged nucleosome binding chromosome for nucleosome, for example.
on the yeast, this is so compact, 70% of the genome sequence. And this definitely do see, we see on the large scale variations in occupancy that distinct variations in the genome of base composition.
So do you think that nucleosome position is why one of the regions having a lot of gene or vice versa?
Very interesting. We are actually, so I think there's, yeah. No show.
You guys in the poly-A trad, it change the occupancy position of the non-upstream. Can you?
Yeah, this touch a year and a half. We're trying to measure nucleus. I think we're finally we have a hand. We definitely critical point to doing those experiments.
Interesting. So have you tried the same technology, nuclear frequencies that you predicted for the model?
We have not tried that yet.
Interesting to see what.
Right. Yeah. In a similar way. But.
Okay.